A healthy living cell operates as a highly organized machine, maintaining its internal structure against the chaotic forces of thermodynamics. Survival depends on a hidden threat, lysosomes. Packed with lethal enzymes, the cell uses two safeguards. First, enzymes are imprisoned behind an impenetrable lipid bilayer wall. Second is a chemical safeguard. They only function in an acidic environment, becoming inert in the neutral cell interior. Survival relies entirely on keeping these internal destroyers perfectly contained. The cell is a ticking bomb, disarmed only by its own delicate chemical balance. The countdown starts at T0. When an organism dies and oxygen flow stops, the cell enters a desperate state of anaerobic glycolysis to survive. This process produces acid as a byproduct, which begins to flood the interior of the cell. This timeline graph shows the chemical collapse. As acid accumulates, the internal pH plummets from a safe 7.4 toward a dangerous 5.0, breaking the cell's second biological safeguard. Simultaneously, escaped enzymes called lipases begin attacking the physical lipid walls of the lysosomes from the outside in. By T+20 minutes, the dying cell has inadvertently manufactured the exact acidic environment required to arm its own destroyers, guaranteeing its impending destruction. At T+20 minutes, the degraded lysosomal membranes finally fail. They rupture simultaneously across the tissue, spilling their contents directly into the cytoplasm. Because the surrounding environment is now highly acidic, these spilled enzymes are instantly activated rather than neutralized. This initiates autolysis, the process where the cell aggressively digests itself from the inside out. In the initial wave, enzymes ruthlessly cleave structural proteins into useless peptides and strip crucial lipids away from the remaining membranes. As the enzymes sweep through the cytoplasm, the physical result is catastrophic, liquefying the internal organelles and collapsing the cell's architecture. Within 20 minutes, the outer structures have been entirely compromised, clearing a path for the enzymes to advance toward the cell's core. The transition continues at T+40 minutes, when specialized enzymes called endonucleases breach the final cellular fortress, the nuclear envelope. This breach exposes the cell's genetic archive, containing the DNA that instructs all biological life. The archive's destruction begins with pycnosis. RNA synthesis halts, and the chromatin condenses into a tight, dense, dark mass. Next, during karyopyresis, enzymes make vicious double-stranded cuts directly into the DNA, severing the genetic code. Visually, the dense nuclear mass fragments and scatters across the ruined cytoplasm like shattered glass. Finally, karyolysis takes over. Extensive enzymatic hydrolysis completely dissolves the shattered DNA into basic building block nucleotides. In destroying the nucleus, the cell completes its structural collapse and systematically wipes its own genetic hard drive. At exactly T+60 minutes, the biological violence finally ceases as the enzymes run out of structures to destroy. All that remains under the microscope is a completely dissolved ghost-like silhouette of a cell membrane. What began 60 minutes ago as a highly organized, thermodynamically improbable system has officially devolved into a disorganized bag of loose amino and fatty acids. Biological death at the cellular level functions as a highly active, heavily armed, and irreversible self-destruct protocol.